Pollination. Pollination is the transfer of pollen which contains the male sex cells from an anther part of the male reproductive organ to a stigma part of the female reproductive organ. This process precedes fertilization. Pollination may occur within the same flower, self-pollination, or between flowers on separate plants of the same species, cross-pollination. In most plants, pollination is carried out either by insects, entomophilous pollination, or by the wind, anemophilous pollination. Less commonly, birds, bats, or water are the agents of pollination. Insect pollinated flowers are typically brightly colored, scented, and produce nectar on which insects feed. Such flowers also tend to have patterns that are visible only in ultraviolet light, which many insects can see but which humans cannot. These features attract insects which become covered with the sticky or hooked pollen grains when they visit one flower and then transfer the pollen to the next flower they visit. Wind pollinated flowers are generally small, relatively inconspicuous, and unscented. They produce large quantities of light pollen grains that are easily blown by the wind to the other flowers. Reproductive structures in wind pollinated plant. Sweet chestnut, Castanea saliva. Female. Peduncle, inflorescence stalk, bracts, leaf-like structure, petiole, leaf stalk, female flower. Prominent stigma protrudes from flower, flower bud. Male. Anther, filament, peduncle, inflorescence stalk, male flower. Part of male catkin inflorescence adapted for wind pollination. Reproductive structures in insect pollinated plants. Micrograph of carpels, female organs. Yellow wort, Blackstonia perfoliata. Stigma, style, boundary between two fused carpels. Each carpel consists of a stigma, style, and ovary. Micrograph of stamens, male organs. Common Centorium erythrae. Calyx, wall of sepals, stamen, anther, filament, pollen grain, endothecium, pollen sac wall. Dehiscid, split open pollen sac. Micrographs of pollen grains. European field elm, Ulmus minor, pore, exine, outer coat of pollen grain. Justicia aurea, exine, outer coat of pollen grain. Corpus, furrow-shaped aperture. Meadow cranus bill, Geranium pratense. Pore, exine, outer coat of pollen grain. Baculum, rod-shaped structure. Box-leaved milkwort, Polygala chamaebuxus. Equatorial furrow, exine, outer coat of pollen grain. Colpus, furrow-shaped aperture. Mimulopsis solmsi, pore, exine, outer coat of pollen grain. Theseum alpinium, exine, outer coat of pollen grain. Trillet mark, development scar. Ruellia grandiflora, exine, outer coat of pollen grain. Columella, small column-shaped structure. Chrysanthera nilotica, tricolpate, 3 colpae pollen grain. Colpus, furrow-shaped aperture. Exine, outer coat of pollen grain. Insect pollination of meadow sage. 1. Bee visits flower with mature anthers but immature stigma. Sepal, labellum, lip forming landing stage for bee. Immature, unreceptive stigma. Anther pushed onto bee's hairy abdomen. Pollen grains from anther stick to bee's abdomen. 2. Bee flies to other flowers. Pollen grains attached to hairy abdomen. 3. Bee visits flower where the anthers have withered and the stigma is mature. Mature receptive stigma touches bee's abdomen, picking up pollen. Long style curves downwards when bee enters flower. Sepal. Labellum, lip forming landing stage for bee. Sunflower under normal and ultraviolet light. Normal light, central area of disc florets, ray florets, ultraviolet light. Paler outer part of ray floret, darker inner part of ray floret. Insects attracted to darkest central part of flower which contains nectaries, anthers and stigmas. St. John's wort under normal and ultraviolet light. Normal light, stamen, filament, anther, stigma, ovary, petal. Ultraviolet light, dark central area containing nectaries, anthers and stigmas. Honey guide directs insects to dark central part of flower.